Hi, AP Bio. So I'm going to go through the agenda for today. So first, it's going to be some announcements. Then I'm going to do a mini lecture on Unit 2, just like I did one on Unit 1 from last lesson. Then in the second video, I'm going to do a mini lecture on Unit 3, and then I'm going to describe the homework for today. So some announcements. First, I've decided I'm just going to leave a link to the calendar I'm using to plan out my lessons right there so you can see what's coming up. Also, just a reminder, it has some links to review resources in it, so I would encourage you to check that out. Also a reminder that today, if you're watching this on Thursday, we have a Zoom session at 1:30, and I hope you join. The link to join is right here. It's also on Google Classroom. And during that Zoom session, I'm going to go over some questions regarding the new format of the AP exam. And also right here, there's a link to the Something I Miss About You Google Doc that I created. So that should take you to a Google Doc that looks something like this, but will hopefully be more filled in by tomorrow morning when you guys are seeing this. And I've got to say, guys, you exceeded my expectations and made me really happy with the lovely things you wrote about each other. It brought me so much joy. So thank you. And then last announcements, just some AP review tips. Just a reminder that there are daily lessons at 1 p.m. every day from College Board, but I wanted to spotlight two of them. These two are links to two of their lessons that go over the content from our Unit 2. So one of them, loading. So this one hasn't happened yet, but it is, this one has happened, and it's about macromolecules. And then this one is, I think, the one, never mind. This one has also happened. Sorry, got myself confused. Anyway, they both happened. Oh, I think one happened today after I planned, but before I record this video. Hilarious. Anyway, okay, but this one is, both of them are about different types of macromolecules, which are topics from our Unit 2, and I think it would benefit you to view them. And then this, which is the first link you saw me pull up right here, is a review on cell structure and function, which is from our Unit 1. So that would also be really useful. You'll see this is quite a long review, very in-depth. So, you could choose to watch part of it, not all of it, or like maybe watch part every few days. One thing I wanted to clarify is, as you've noticed, I've been saying our Unit 2 versus AP's unit numbering system, which is a little different. So I wanted to just clarify that for you. This shouldn't really make any difference for you, but just if you're seeing reference to things, I wanted you to rest assured that it's all figured out. So here are the units that we did. And so in my last video from Tuesday, I did a mini lecture on our Unit 1, which was called Cells, Structure, Transport, and Reproduction. And that correlates to the AP College Board Unit 2 on cell structure and function, and also a little piece of their Unit 4. Today, I'm going to be doing a mini lecture on Unit 2 on molecules, energy, and metabolism, and also on Unit 3, cell communication. Our Unit 2 corresponds to two of their units. So this was probably our biggest, our meatiest unit of the year. It corresponds to AP's Unit 1, Chemistry of Life, and AP's Unit 3, Cellular Energetics. Our Unit 3 on cell communication correlates to AP's Unit 4, cell communication, minus the part on cell cycle, which we covered in our Unit 1. And then you'll see that our units 4, 5, and 6 from this semester correlate pretty cleanly to the AP units 5, 6, and 7. So that's just a little like language clarification for you. But now, for the rest of this video, I'm just going to do a little lecture on Unit 2 to remind you of what we talked about. This was right around September, October time. So we started this unit by talking about macromolecules. Remember, there are 4 types of macromolecules: nucleic acids, carbohydrates, lipids, and proteins. And we talked about how they are made up of different building blocks. Oh, by the way, you probably want to take notes. So you should pause this right now, go find a lined piece of paper, back it up a bit, listen to that again, and then keep watching. One little thing, I've included my image sources in the notes of the agenda, and I would highly recommend checking them out, especially this one, because it's actually a children's book that I think would be really cute for you to see. It's probably someone's AP Bio project from another class, but it goes through the different 4 different types of macromolecules and what makes them each different from each other. Then we zoomed in specifically on proteins because proteins are actually one type of protein are enzymes. And we spent a whole bunch of time talking about enzymes. And remember that proteins have 4 layers of structure. So it starts out, remember we all stood in a line, that's the chain of amino acids, primary structure. Then there are kinks, that's secondary structure. Then it folds up, that was when you guys, people with white shoes, found other people with white shoes. So that's tertiary structure. And then quaternary structure is if we have another clump come in and join. And this folding pattern might seem pretty random, but it's actually super precise, and we're going to get to that in a second. But basically, enzymes, remember, they bind with the substrate, sort of like a puzzle piece, and they make a reaction happen faster and with less energy. So here is if there's no catalyst, meaning no enzyme. Here is if there is an enzyme. So it requires less energy if there is an enzyme present. Then we did our enzyme lab, and here's a little image I found from someone's online lab report. But remember, you guys, your independent variables were different. Some of you, it was the amount of catalase, that was the enzyme. Some of you, it was the amount of hydrogen peroxide, that was our substrate. Some of you, it was the pH of the solution. Some of you, it was the temperature of the solution. And you were all measuring, though, the amount of bubbles, amount of oxygen gas produced, and the rate of how that was produced. Experimental constants, everything that was not one of the independent variables, and the control group, remember I did a demo, and that was your comparison point. And what we were looking at was that if we change one of these things, it's going to change a graph of the enzymatic activity. So especially we looked at pH and temperature and how all enzymes have an optimal pH and an optimal temperature, meaning the pH or temperature at which that enzyme works best. And that has to do with what we talked about on this previous slide, the folding pattern of that protein. Because the enzyme is going to fold differently if it's at the wrong pH or the wrong temperature, which then causes it to denature and then causes it not to work as well or at all, potentially. Then we moved on to talking about photosynthesis, which of course is made possible by many, many enzymes. There is a lot of detail involved in the process of photosynthesis, but at its most basic, photosynthesis has to do with taking sunlight's energy and using that to make glucose. So the sunlight's energy, remember, comes into the light reactions, breaks water. That breaking of water releases some energy. That release of energy is used to make ATP through chemiosmosis. That ATP then is used to fix carbon dioxide into a sugar molecule. That's really the goal of photosynthesis. Oxygen is released as a byproduct of breaking the water. If you don't remember much about chemiosmosis, this would be something to go more in depth on. I'm not going to go in a lot of depth on it here, but I am going to remind you that it comes up here and again in cellular respiration. And remember, in both cases, chemiosmosis has to do with having a proton gradient. So a lot of protons or hydrogen ions on one side of a membrane. And the only way they can get through that membrane is by going through ATP synthase. And ATP synthase then makes ATP when those protons go through it. And so it is the imbalance of protons on one side versus the other that actually generates the power to make ATP. And so that happens again at the end of cellular respiration, because it's at the end of cellular respiration that ATP is formed. It is the goal of cellular respiration is to make ATP. And this, so in summary, what cellular respiration is the process of taking glucose in and using that to make a whole bunch of ATP. There's three main parts of cellular respiration. We start out with breaking the glucose into pyruvate. That happens during glycolysis, which is outside of the mitochondria. Then we go into the mitochondria. We have the citric acid cycle, which is when the carbon dioxide is released from breaking apart the sugar molecule. And then we do chemiosmosis as part of oxidative phosphorylation to produce a whole bunch of ATP. Again, this is all super, super in brief. We're going to be reviewing a lot more over the next few weeks, but just wanted to give you an overview right now to remind you. And then last slide for Unit 2, we did our lab on photosynthesis and cellular respiration. If you remember, we were using algae beads and you were using a pH indicator solution that changed color based on pH. And so different groups had different independent variables. The ones I remember, some were looking at different amounts of light or different qualities of light, meaning sunlight versus a lamp. Some were looking at the wavelength of light. So remember, if you have different colors of light, that's different wavelengths of light. So they were looking at the impact of that because photosynthesis can only happen in certain colors of light. It can't happen in green light, which is why plants are usually green, because chlorophyll reflects green light and doesn't absorb it. The dependent variable was the change in pH, which we were looking at by looking at the different colors. And the reason why we were looking at the change in pH is because if there is more carbon dioxide, the solution will become more acidic. And if we look at how much carbon dioxide levels are changing, that tells us how much photosynthesis or cellular respiration is happening, which is what this is showing here. So when the solution was super yellow, that meant it was super acidic. And so that meant that cellular respiration was happening because there was a lot of carbon dioxide being put into the solution. When the solution was really purple or blue, that meant it was very basic, which meant there wasn't much carbon dioxide, which meant it was being used to do photosynthesis. And that was what we did with this lab. And that was a super, super brief overview of Unit 2. Again, I know that was crazy fast. That was a really challenging unit. So feel free to watch this video multiple times. Also know that I do have videos, if you remember, on photosynthesis and cellular respiration already on my YouTube channel. And don't worry, we're going to be reviewing these a whole bunch more in the next coming weeks. This is just a preview. Okay. So now I'm going to do my second video on Unit 3 once I figure out how to pause the video.